The Gulf of Maine is a very productive ecosystem. It's home to right whales, bluefin tuna, and lobsters. But at the foundation of this ecosystem are plants that are so small and uncharismatic, they're often taken for granted. But here at the Bigelow Laboratory for Ocean Sciences, phytoplankton takes center stage. I meet Bigelow researchers Nicole Poulton and Greg Gavelis walking down to the dock to get a water sample to demonstrate their process. All right, so Nicole is grabbing a sample off the end of the dock. What's she doing here?
Well, she's taking a sample of water from a predetermined depth, and the nice thing about that device, that Niskin bottle, is she can lower it down to the exact depth that she wants and then send that little weight down, and it triggers kind of a trap door that traps a little bit of water there. So she's a nice representative piece of the ocean at that exact point that we're interested in studying.
The water looks clear, but Poulton says that's deceptive.
It looks clear. However, in every single milliliter in here, we have over millions and millions of bacteria. So, in addition to that, hundreds of thousands of phytoplankton. But the density is still not dense enough that you would see them with the naked eye. You have to use a microscope to be able to see them.
Up at the lab, Poulton uses vacuum pressure to pull the water sample through a fine glass filter. After filtering the water, Poulton has a collection of phytoplankton and other small organisms tinting the filter green. Wow.
Yeah.
So everything's in there.
Mm-hmm.
It's just all mushed up together.
It's all mushed together on top of this small filter. So this filter here has really small pore size, so it allows all of that bacteria to accumulate right onto the surface. The only thing that does pass through tends to be viruses because they're so small.
By closely studying plankton, Bigelow researchers have made important discoveries and published them in major scientific journals. One of the most dramatic and frightening discoveries made at Bigelow, that phytoplankton in the Gulf of Maine have declined by 2/3 over 20 years. And then there was another discovery here, published in the journal Nature, that took the oceanography world by storm. Poulton and colleagues studied the most abundant plankton, bacteria called Prokaryoplankton, and showed that in the Gulf of Maine, less than 3% of these bacteria consume up to 30% of the oxygen in the ocean, while they're also known to produce a large amount of CO2. This all may seem wonky, but it's important. It shows that some microscopic organisms have an outsized influence on the planet's climate.
So this is kind of the dark side of the picture with phytoplankton. When we think about phytoplankton, we think about tiny plants or algae, and they make 50% of the oxygen we breathe. But then there's all these other members of the plankton, especially bacteria, that are breathing a lot of that oxygen immediately. Nature is all about balance. And so we have areas of the ocean, like kind of out here, off the dock that we were just looking at, where it's nice and green and vibrant and it's creating a lot of oxygen. But then we have other areas where there's actually more bacteria than algae or more active bacteria that are breathing up so much oxygen that it's formed dead zones where not even fish can survive.
Not only do phytoplankton produce half of the oxygen on Earth, they're also the tiny building blocks of massive creatures like whales.
But it's the base, the microscopic base of the food chain, which is both the most important and also the hardest to see. So any method that we can develop that helps us understand that better is a huge step into understanding how the ocean as a whole works. And single-cell genomics is, I think, an important part of that.
There's a laser beam.
To understand plankton on the single-cell level, Poulton uses something more sophisticated than the bulk sample she caught in the filter. It's called a flow cytometer. It sends a stream of tiny cells flowing in single file past a laser beam, which allows researchers to identify them.
The thing is that so it's thousands of going past that laser beam, but they're all going down in single file, and the laser beam's intersecting them, and we're capturing all that data right here at this point.
Yep. This precision technology is often used in the biomedical field, but Poulton says it's equally important for understanding the world's oceans.
One of the big issues is that a lot of people don't realize the importance of these organisms. They're what we call the base of the food chain, and if we don't have those organisms, then we don't have the megafauna that we're used to, the fish that we consume, and the upper trophic levels.
So no menhaden, no seals, no bluefin tuna.
Exactly.
None of that.
None of that.
And Poulton and Gavelis say it's more critical than ever to understand the microscopic plankton that are not only feeding fish and lobsters and whales, they're also producing half of the planet's oxygen. For Borealis, I'm Murray Carpenter.